Good morning, good afternoon, good evening, everyone. Wherever you're joining us from today, welcome. I'm Roger Peck, Associate Director of Diagnostics at PATH, and I'll be your host and moderator for today's webinar, Biobanking for Pandemic Preparedness. We can jump right into the slides, so next slide, please. In the best of times, biobanking is a complex topic. During a pandemic, time is of the essence, and resources are often stretched. Advanced planning and preparation are needed to efficiently and effectively respond. As we prepare for pandemic biorepositories, we acknowledge existing resources, leveraging previous and current work, and strengthening partnerships as we right-size our responses. A successful biospecimen repository is multifaceted. Considerations must be given to the statutory requirements, standards, and best practices. These are important to assure beneficence and equity for the individuals and populations from where specimens are sourced. They assure that specimens and associated data are of high quality and relevant, and they assure that specimens can be accessed. We then layer on the emergency and use cases related to operational context, whether we are in normal surveillance mode or in situations of elevated importance, such as outbreaks, epidemics, and pandemics. Next slide. Today's webinar will highlight only a few of these topics and just scratch the surface. We hope to engage with you and the broader community to hear your comments, questions, and areas for further discussion and future engagements. So if you're joining us live, welcome. Please submit your questions to the panelists. However, we will not answer questions in real time, but instead we'll follow up in the webinar report that will be available to you after. If you're joining the webinar from a recording, our contact information is included at the end of the presentation if you would like to follow up with us. Now, let's dive into the webinar topics. As you may have seen, CEPI and PATH recently announced a partnership for building a biorepository framework that is aimed at addressing unique challenges of operation during disease outbreaks. Dr. Salorm Avamega, project lead and scientist at the Coalition for Epidemic Preparedness Innovations, will introduce you to the Biospecimen Sourcing Initiative. Salorm, over to you.
Yes, thank you very much, Roger, and hello, everyone. Once again, thank you for joining today's Biobanking for Pandemic Preparedness webinar, the first of many webinars to come under this initiative. Yes, and I would love to introduce you to the CEPI's BSI project. And as Roger said, there has been blog posts on this initiative on almost all social media platforms, which is exciting. And I will let you know how this fits into the 100-day mission. Next slide, please. And next slide. Yes, and a bit about CEPI. So the Coalition for Epidemic Preparedness Innovation is a global partnership with a mission to accelerating the development of vaccine and biologic countermeasures against epidemic and pandemic threats. You see, CEPI envisions a world where epidemic and pandemic are no longer a threat to humanity. So our 2022-2026 strategy, also known as CEPI 2.0, aims to enable the creation of new vaccine within 100 days in response to emerging threats when appropriate. This is being done through the Prepare, Transform, and Connect strategy. And to achieve this ambitious 100-day mission, it means vaccine candidates should ideally be available within the first 30 days to push it through large-scale manufacturing. And the time to source for appropriate biospecimen for assay development is even shorter, hence the need for this initiative. Next slide, please. And biospecimens are crucial in product development, especially for reference material, for creating reference material. High quality reagent and standards are necessary resource for developing new vaccines that meet user requirements. These materials are needed at the beginning of product development. However, these materials take a long time to source, verify, and make available for novel pathogens particularly those affecting hard-to-reach population. It is therefore important to have a globally accessible resource with pre-established and vetted processes for sourcing, verifying, archiving, and distributing biospecimen to support the development of vaccine during novel pathogen presentation. Next slide, please. CEPI has already been collaborating with various partners for biospecimen sourcing to support the development of WHO international antibody standards for our priority pathogens and disease X. And lessons learned has been what has led to this initiative. Next slide, please. So the BSI, the main objective of the BSI is to facilitate timely access and ethical sourcing of biospecimen in an outbreak to support assays and vaccine development for the 100-day mission. Next slide, please. And BSI is being set up in 2 phases. Phase 1, governance structure, a framework and guiding principle for BSI operationalization. And this is actually ongoing, and we hope to finish it by June 2026. And phase 2, BSI implementation, involving institutions and initiatives that we can collaborate with to support timely access to biological materials of interest to support the 100-day mission. Next slide, please. And for the phase 1, we are collaborating with PAD, and PAD is leading the establishment of the governance structure and library of guiding principle and document for scientific readiness. Next slide, please. And we plan to launch a call for proposal summer 2025. And we hope that when that call is out, many of you will express interest to join this initiative and collaborate with us to pull this through. Next slide, please. So how does CEPI envision BSI goals being achieved? CEPI's aim is not to reinvent the wheel, but help oil the wheel in collaboration with global efforts to support timely access. To that end, we hope to support a governance and management framework for BSI operationalization, pre-establishment of standardized protocols and methods for scientific readiness, partnership with institutions with capacity to develop reference standards, collaboration with biobanks and biorepositories for long-term storage. We know there are many stakeholders and subject matter experts in the field, so establishment of an external advisory group is critical, who will advise on key operational priorities. And sustainability is key. And so regionalized business model and ownership is something that we will aspire for. Next slide, please. What will success look like? By the end of phase 1, we anticipate having published governance and management framework to support BSI operationalization. We hope to have an active external advisory group to provide guidance on key operational priorities for the BSI. Develop and validate library of guidance document that support BSI operationalization by articulating guiding principles. Then phase 2, we hope to have established an active network of global biobanks and biorepositories with other initiatives to advance scientific readiness for outbreak response. Next slide, please. Biospecimen this project is actually a complex and a huge undertaking. And we rely on all the stakeholders here having joined us today to really pull this through. And together, we know it will be a success. Next slide, please. In summary, timely access to biospecimen and sharing are indispensable to fulfilling the 100 days mission, not only from a technical standpoint, enabling rapid diagnostics, therapeutics, and vaccine R&D, but also a matter of global equity, governance, and system resilience. Without a streamlined, secure, and inclusive specimen sharing framework, the mission's ambitious timeline and impact will be undermined. Next slide, please. And with this, thank you for listening and hope to see you on the other side. Thank you.
Lauren, thank you so much for sharing that perspective. Next, I'd like to introduce Austin Liu, who is a health policy analyst at the Coalition for Epidemic Preparedness Innovations, who will speak to CEPI's perspectives on the pandemic agreement, which has been a topic of discussion at this year's World Health Assembly. Austin, over to you.
Thank you, Roger. Thanks for having me in this webinar. CEPI has the pleasure to actually present our perspectives on the pandemic agreement when it's still being negotiated in another PATH webinar. So we are really pleased to be here as the WHO member states adopt the pandemic agreement earlier this week at the World Health Assembly. If you go to the next slide, I think through my presentation, I would aim to explain our rationale for engaging in the pandemic agreement process, to share our perspectives on some key elements that have been adopted in the pandemic agreement, and also to outline what are the next steps. Next slide, please. So why did CEPI engage? I think, as Salom, my colleague, mentioned, 100 days mission and equitable access are the North Star of CEPI. And to achieve that, there are 4 capabilities that we think are needed to ensure a resilient and equitable global preparedness and response ecosystem in order to respond to epidemic and pandemic threats within 100 days to save lives and minimize global disruption. And for that, we need the ability to detect, to characterize, and respond to emerging infectious disease threats, to be able to rapidly design and develop product and have clinical trial materials and test vaccines. Third, to be able to rapidly produce products, scale up and roll out vaccines. And finally, to approve vaccines rapidly through pre-agreed pathways and get vaccines into initial use. Next slide, please. So last year, WHO member states agreed on amendments to the international health regulations, and we welcome that. Those amendments include, for example, the promotion of equity and solidarity, the obligation to remove barriers to timely and equitable access to health products, the authorization for the WHO Director General to be able to share product dossiers with regulatory agencies subject to the manufacturer's consent, and to establish the coordinating financial mechanisms to promote sustainable financing. However, to build a PPR ecosystem that can realize our common goal of developing a safe and effective vaccine within the first 100 days following the emergence of a pandemic threat, and ensure all countries have rapid access to the tools required to stop the outbreaks from becoming pandemics, the pandemic agreement is a once-in-a-generation opportunity. Next slide, please. So at the Intergovernmental Negotiation Body, the INB, CEPI has offered 2 levels of representation. First, CEPI participated as a relevant stakeholder in the INB. And second, CEPI also, through leadership and other channels, participated in experts consultation in the pandemic agreement discussions. And we continue to demonstrate through practical examples to catalyze actions for the 100 days mission and equitable access through the CEPI programs. In the INB, we highlighted 3 key policy asks throughout the process. First is to sustain preparedness investment in R&D, and to ensure that government R&D funding agreements include contractual requirements to support equitable access with terms such as licensing, affordable pricing, technology transfer, information sharing, and equitable allocation. Second is to support a network of partnership for end-to-end collaboration of medical countermeasures, development and response, and to promote investments in economically sustainable, geographically distributed vaccine manufacturing facilities with the capability for rapid response. And finally, to establish a multilateral system that can support rapid and efficient sharing of samples and data on pathogens with pandemic potential in order to expedite R&D for medical countermeasures at all the times, as well as the equitable access to the outputs of such R&D. Next slide, please. So in the following slides, I'm going to share some CEPI perspectives on some key elements in the pandemic agreement. Next slide, please. So a number of articles relate to medical countermeasure development, manufacturing, and access. So I will highlight a few today. If you go to the next slide, starting with Article 4, Pandemic Prevention and Surveillance, it requires countries to prevent the accidental exposure, misuse, or inadvertent release of pathogens and ensure the safety and security of transportation and cross-border transfer. For example, in Article 6, about preparedness, readiness, and health system resilience, it requires countries to sustain and monitor and strengthen health systems for laboratory and diagnostic capacities, such as through the application of standards and protocols, including for laboratory biological risk management, and to support regional and global networks. Next slide, please. Going to Article 9 on research and development, which is what CEPI's mission is focused on. Article 9 requires countries to sustain investment in research institutions and network that can rapidly adapt and respond to R&D needs in the event of a pandemic. And it requires them to accelerate innovative R&D consistent with biosafety and biosecurity regulations. It also promotes geographically diverse and sustainable R&D capacities with a strong emphasis on collaboration and rapid sharing of results from research during pandemics. And finally, one of the most powerful mechanisms that we believe and we have been advocating on towards equitable access is Article 9.5, which requires each country to develop and implement policies regarding the inclusion of equitable access conditions in publicly funded R&D agreements, including terms such as licensing and tech transfer. As we don't know where the next innovation will come from, it is important that every country includes this kind of equitable access provisions. And if every funder includes these conditions, then it would strengthen our collective ability to promote equitable access. Next slide, please. So going to Article 10 and 11 about local production and technology transfer, there also are the provisions that after R&D to enable equitable access, that includes requiring countries to take measures to achieve more equitable geographic distribution and rapid scale up for the global production of pandemic-related health products, to promote and incentivize public and private sector investment, including purchasing arrangements and partnership to expand manufacturing, as well as establishing long-term contracts for pandemic-related health products. And in Article 11, it includes requirement for countries to promote technology transfer, which may include licensing, capacity building, relationship facilitating, or other incentives or conditions linked to research and development. And it also requires countries to enhance the availability of licenses on a non-exclusive, transparent, and broad geographic basis that can enhance the production of health products like vaccines. Next slide, please. And finally, Article 8 on regulatory system strengthening calls on countries to strengthen national and regional regulatory systems to ensure timely, safe, and effective approval of pandemic-related health products, to reduce delays in authorizing vaccines and other tools during crisis, including support for expedited regulatory review or emergency regulatory authorization, adopting regulatory reliance mechanisms, aligning relevant technical and regulatory requirements, and strengthening national regulatory authorities and regional regulatory systems capacities. So there are, across these provisions and articles, there are various mechanisms and provisions to accelerate R&D for the 100-day mission and to enable equitable access to the outputs of the R&D. If you go to the next slide, and the next slide, what is next? WHO member states at the World Health Assembly adopted the resolution, and they will agree to draft and negotiate a pathogen access and benefit sharing system, PAB system, through the intergovernmental working group that is being set up. In the pandemic agreement, they agreed on some principles for the PAB system, which includes commitments to rapid and efficient sharing of samples data of pandemic potential pathogens, which is important to expedite research and innovation. From CEPI's perspective, a multilateral is necessary to mitigate possible delays caused by bilateral access and benefit sharing transactions on pathogen samples and data. Moreover, interoperability and speed are also critical in facilitating seamless data exchange across many databases and data types, and to combine and compare data from various sources for vaccine development. In the pandemic agreement, in Article 12, there are also some other benefit sharing measures that are agreed, such as targeting 20% of the real-time production of vaccines, therapeutics, and diagnostics of participating manufacturers in PABs, and also other sharing provisions such as royalty-free, non-exclusive manufacturing licenses to manufacturers. But this will be, the details of the systems will be negotiated in the Intergovernmental Working Group. Next slide, please. And this is the final slide. In summary, while we should celebrate the adoption of the pandemic agreement, we must also recognize that the agreement on its own will not deliver the level of pandemic preparedness and the world urgently needs. It will take sustained investment, enduring political commitment, and unprecedented scientific collaboration to create the systemic change. For CEPI, one of the most powerful mechanisms to enable equitable access is through Article 9.5, which requires countries to implement policies regarding the inclusion of equitable access provisions in publicly funded R&D agreements. But CEPI stands ready to work with countries and our partners to share our experience and to demonstrate practical examples to catalyze actions for the 100 days mission and for equitable access. And the webinar on this BSI Biospecimen Sourcing Initiative is an example of how we are catalyzing these actions for the 100 days mission and equitable access. And finally, countries do not need to wait until the ratification of the agreement to implement relevant approaches such as Article 9.5 to support equitable access and the 100 days missions. Thank you. Back to you, Roger.
Great. Thank you so much, Austin, for that wonderful summary of what's happened with the pandemic agreement so far and what the next steps are. I'd like to move on to Daniel Simeon-Dubach, owner of MedService, who will share key insights on developing respectful partnerships and how to align stakeholder goals within the overall aims of biobanks. Daniel, over to you.
Thank you very much, my friend, and thank you for inviting me to this webinar. It's really exciting. As you can see, the title of my talk is relatively long. In this presentation, I will discuss partnerships between different stakeholders and biobanks and how to align these goals with stakeholders and the aim of the biobanks. To better understand the topic, I would like to first talk about some definitions. Can I have the next slide? So what is the aim of a biobank? Well, most obviously, it's preserving biological resources and data. That's clear. That includes also rare samples, of course, and specific disease cohorts. And one should not forget to establish a reference collection that includes also donation from healthy donors. Another aim is facilitating and support medical research. Here it's really important that these data are of a defined quality that fits the purpose for the specific research. By doing so, it can accelerate precision medicine and also promote scientific collaboration between different research disciplines. It also improves public health by contribute to disease surveillance and epidemiological research, support development of diagnostic tests and preventive strategies. Data integration is very important also, and a lot of biobanks are doing that quite successfully nowadays. And last but not least, ethics is key. And all the biobanks operate under strict ethical guidelines to protect the privacy and rights of donors. Can I have the next slide? So on the stakeholders of biobanks, there are, as you can see here in this slide, for me, there are primary and secondary stakeholders. The primary stakeholders, of course, the core stakeholders are sample and data donors, so the participants. This is for them, the consent is really the key, and they need to trust that biobanks are executing what they are doing in a ethical, correct way. The biobank staff is also key for a biobank. Without the well-trained staff, you cannot run a biobank successfully. And then certainly the research scientists, they come from academia, from clinics, from industry. Doesn't really matter from where. They are also as important as the others. Funders, of course, without funders, you cannot run a biobank. A biobank as a small or even medium-sized enterprise that is meant to last for long term needs to have a strong focus on the funding and financial sustainability. The hosting institutions play a critical role to provide the infrastructure and oversight and institutional support. And again, ethics, or specifically the ethics committees, play a very important role. Then the secondary stakeholders, there are the regulatory bodies that do more than just ethics, the healthcare system in general that benefit from the research findings and improve that they will profit from the improved clinical care and also from the reputation. Industry partners, of course, very important, and patient advocacy groups. You have to talk to these patients, otherwise you're not included in what is going on. The general public also can profit a lot from the biobanks, and the biobanks need to pay attention to them. And then last but not least, the future generation, because they will ultimately benefit from long-term biobank collections. Next slide, please. So how to align the goals of the biobank stakeholders with the overall aim? For me, there are four pillars, and one is the strategic approach. The second one is more the operational alignment, and that's from Roger. Okay, and so now I cannot see the slides because okay, and then communication and engagement and some practical implementation tools. Next slide. So when you see here a little bit more in detail what I meant with these different these four government pillars, it's the strategic governance, it's the clear vision, mission, and value, and I'll come back to that. It has to have an inclusive governance structure with advisory boards that includes participants, scientists, community members, and so on. And also a decision making that includes all the stakeholders. In the operational alignment mechanism, transparency, sharing initiatives, and we heard about that, are key. There are the FAIR principles is one of the other initiatives that are very successful. It's important to have a well-established benefit sharing model, and of course it's a sustainability model that focus on operational, financial, and sustainable dimension of sustainability. The communication, again, it's so critical to talk to all the stakeholders, educate them. They will love to hear from what biobanks are doing. But also feedback mechanism and evaluation mechanisms are important. So you learn what your stakeholders think and what they need, and engage the stakeholders. Again, they're very happy to help you. Some practical tools. There are some stakeholder-specific value proposition. There's also the adaptable governance, and one topic that people don't really like to talk about is how to resolve conflicts. Because when you collaborate with people, there's a high potential for conflicts, and you need to solve that, because otherwise you cannot do that. Next slide. So I told you the business planning is really key, and I think if you are new in the area as a biobanker, or if you're an established biobank, you should have a business plan, and the business plan includes 4 key elements. One is the vision and mission. Without that, you cannot achieve what you want to have. You can be, this can be changed all the time. You need to do a SWOT analysis. Yes, I'm just, this is the last slide, Roger. Thank you. You need to do a SWOT analysis. I can tell you now, just before this call, I have, with another business plan I'm involved in, I looked at the SWOT analysis and readjusted it. You need to have SMART goals and metrics, and you need to do a risk management, a risk assessment. By saying that, I showed the last slide, which I thank you very much for your attention, and thank you for having me had this webinar.
Great, Daniel, thank you so much. Really appreciate your perspective on stakeholder and partnership engagement. I'm sure topics we can discuss for hours. Next is Dr. Zisis Kolozdikis, Head of Laboratory Services and Biobanking at the International Agency for Research on Cancers, World Health Organization. Zisas will share his experiences and perspectives implementing rapid specimen collection initiatives. Zisas, over to you.
Yeah, thank you very much, Gregory. Next slide, please. So the discussion that we initiated at the agency with regards to the rapid collection came as a result of the meeting we had at the G20 during the pandemic. And that was driven by the data primarily, and that's why we have guidelines for data. But very quickly, we realized that this has to be something bigger. Next slide, please. So 2 years later, in the G20 task group with Indonesia, we started putting a little bit more detail on what does it mean, how does public health interact with digital health, with the data, with the laboratory settings. And next slide, please. And as a result, we managed to produce a policy brief on how you build back better. And this was very much focused on the laboratories, and specifically the laboratories that deal with infectious disease outbreaks. Next slide, please. So we decided that this was a good starting point, but it was very general. And indeed, we had to look regionally for more detail. And one of these regional initiatives that we undertook was to look with the ASEAN Health Secretariat into the different types of biobanking that take place within Southeast Asia. Now, ASEAN is a union of 11 countries, and the member states at the moment include often ASEAN +3 conformation that includes China, Korea, and Japan. But what we find is that in that part of the world, we have very few biobanking activities that are structurally integrated, and we could probably call them mature biobanking activities. Most of them, over 80%, are customized activities. So these are project-driven, event-driven activities, which for the purposes of pandemic is probably okay. But it means that every time we recreate the wheel, there isn't an underlying mechanism to support this when it's needed. And then we have some functional or collaborative sharing of data or samples that's driven by the industry, but this is very, very much limited in numbers. Next slide. So last year, in May of 2024, the health ministers of the ASEAN member states signed an overarching agreement. This is a very high-level agreement, but it sets the tone that we do understand we need to collect some samples. We need to be able to at least collaborate regionally at a minimal level. But this cannot be done just on its own. It has to be supported by training and capacity building. It has to be supported by capacity building when it comes to data, the information sharing and analysis. It has to link to innovation, coordination, and support, and that links directly with what Salome was saying earlier. And it has to be complementary to existing structures. So there is a big step moving forward that we have a common understanding, at least in one region of the world, but we need to put flesh into the bones of this agreement. Next slide, please. One key concern that came up with all of the stakeholders was the protection of the intellectual interest, and this is something that's an ongoing concern. One of the reasons is because a lot of the biobanking activities were set up under the framework of research, and the IP was never part of that consideration. So now when it comes to the sharing of samples and potential commercial use, or secondary use even, that has to be outlined, and the law has to catch up with what the reality needs us to do. Next slide, please. But I don't want to be very critical. So when we map the development of the legal and regulatory framework, you can see that in 2000, so this is when the human genome was decoded, there was very little in terms of the regulation and the activity of biobanking. Next slide, please. If we fast forward 10 years, you can see that there is some activity. Next slide, please. And 20 years later, you can see that the direction of travel is in the right direction. And so I'm very optimistic, but there is space for adaptation. There is still an opportunity for the field to mature. Next slide. So what does the agency do specifically? So IARC has created in 2013 BCNet. This is the Biobank and Population Cohort Network. It's about 50 institutions, or just over 50 institutions in 30 odd countries. These are low middle income countries across the world where we provide research-ready infrastructure network, which means we go in, we do in-person training, we do online support, and we guarantee to some extent that the practice of biobanking is appropriate. So when the opportunity arises, these infrastructures can be onboarded into the activities of the agency or the scientific activities of the time when something is needed. So this is not only driving the capacity, but also the inclusion and representation of samples from those areas into the general research activity. Next slide. So how do we prioritize? So we use a lot of the parent initiatives. So that was an example where on the sidelines of the Lancet Oncology Commission, we use that opportunity to map the biobanking practices in sub-Saharan Africa. And then we start to break down the basic components into smaller components that can drive then initiatives. For example, when we talk about leadership and coordination systems, we really mean that we need dedicated leadership training so that when a rapid response is required, the leadership is aware of what needs to happen and can take that decision fairly swiftly with a full understanding of the next steps that will follow. Next slide. And we do that bottom up as well. So we've done this for biobanks for infectious diseases. So you can see we'll talk about the infrastructure and what are the challenges, what are the enablers, and so on. Next slide, please. So we start getting some granularity. And if you click once, you can see that, let's click. There you go, once more. And once more. So you can see that we have a good understanding now. If we go to most biobanks in most low and middle income countries, what should we see in those freezers, in those cupboards? We have a fairly good understanding. What is really missing is what's on the next slide. And that is our understanding of the data that is there, and more importantly, the quality of the data that's been captured. Next slide. So what do we anticipate? We do anticipate a lot more complexity to arise because biobanking can now service both the collection and distribution of samples and data, and the two do not always have to go together. Next slide. And we also need to anticipate a lot more discussion about the ethics and IP. So the IP we've mentioned earlier, I'm not going to repeat, but in terms of the ethics, we do talk about the implementation of artificial intelligence. Most biobanks in low middle income countries are not ready yet, but the artificial intelligence has to be prepared. It has to become trustworthy so that when they're ready, they can implement, and they're not really that far behind. Next slide, please. So I'll leave it here. These are my contact details, and more than happy to take any questions. As we said, we'll respond to the questions later on. Thank you.
Great, Zisas, thank you so much. Really appreciate those valuable perspectives and insights. Our final presenters are Dr. Mei Chu, Clinical Professor of Interim Director for the Center for Global Health at Colorado School of Public Health, and Dr. Tomas Janesh, Professor at Colorado School of Public Health and Senior Scientist at Heidelberg University Hospital. Mei and Tomas will discuss access and sharing of regional representative characterized specimens in a federated and distributed biospecimen system. Mei and Tomas, over to you.
Okay, well, thank you. Thank you for inviting us to give this talk. Can we have the next slide? And so Oops, can you go back to the first slide? There's two parts to this slide. I'm sorry, I didn't see that. First, I'm going to talk about the 10 by 10 partnership pilot project that we have. And then Thomas will follow up with the need for data harmonization and all the rules and regulations that come from that. So thank you. Next slide, please. So the operating principles of the virtual biorepository system was born in 2019 after the Zika outbreak and of course through the COVID pandemic. And it's an idea that another partner who's here on our panel, Dr. Judith Geary, had conceived for years about a grassroots platform, a platform to help connect contributors and users to meet their needs for characterized quality samples. And we're very lucky to be able to collaborate with FIND and with PATH to have that discussion during the COVID pandemic. And then we saw ourselves as a hub for templates for legal, ethical, standardized procedures and training, and with links to other resources. And then as a forum for discussion and sharing specimens and biorepository challenges and solutions, many of which you've already heard. Then what we would focus on too is provide some services. And we had a vision of providing dashboard and map site for different access to different links and data for sharing specimens, which PATH and FIND and others have already done. And we partner with them. And then we want to make sure that we're aligned and jointly in similar parallel efforts around the world, as we've mentioned already. The last piece that we felt was important was to provide access to reference materials, because standardization and quality control of specimens and use of those specimens is important in developing diagnostics, for which we talked a lot about with CEPI. And so I think many of the partners here are familiar with us. Next slide, please. So the whole concept that this is, instead of a top-down system of creating a system, we ask partners to help us create that system from the different sites and grassroots, so that it's going to be a sustainable, equitable grassroots system for timely sharing of specimens for future outbreaks. And that we wanted to have target representation to samples that reflect global diversity with focus on populations of LMICs. And this is because a lot of our papers, if you go through and look at the work that we do and others do and manufacturers use, most of the background material, or the normal, if you will, come from northern high-income countries. Very few are LMIC derived. And that we want to make sure that this is a distributed system, locally managed, maintained locally. So it's a very federated system under common standards of quality and equitable accessibility. Next slide. So our focus right now is to stand up a pilot project called the 10 by 10. The 10 by 10 is essentially 10 countries that are willing to join us, that will provide 10 mLs of serum or plasma from 10 different normal, healthy individuals representative of their countries and sites. And what we intend to do is, when we finish with this 10 by 10, then we will then expand it to a global system using a buddy system, and that eventually we see this position many places. How do we recruit them? We recruited them through a number of ways, talking to PATH, FIND, CEPI, and others who already have contacts in low and middle income countries, who are already in other networks, and we interviewed them for their willingness to join. And so we aimed for participate, that they already have representation, participation, other sample networks, their peer recommendations, and then we did an extensive round of interviews to gauge interest and willingness. And before doing this, we actually did several rounds of surveys, asking people what their interest might be for a biobank of this nature. And we also did a Delphi process to prioritize what we're going to do. So in the end, the 10 by 10 to be included, you had to have geographic distribution. You have to have biorepository experience, that you know about specimen collection storage already have, access to good temperature, storage capacity. It has been working with inventory and data management, have had clearance through IRB and other informed consent collection, and have shown and had been willing to share and ship specimens. We asked each of our 10 by 10 sites to commit to 2 to 3 years, and then they need to continue to work with us during that period to develop the principles of the VBS. Next slide, please. So we are now funded by the European Commission under Contagio. Contagio stands for Cohorts of Networks to be activated globally for outbreaks. So we are also a preparedness group, and that we have 3 working groups under Work Package 4, and Thomas will talk more about that. And we're talking about governance, organization, as many of you have already mentioned, that's so important to have, and also to make sure that we are working with informed and broad consent, because this is a very critical part for acquiring specimens. Then we are going to map regulatory requirements, and ethics is very important, as many of you have already talked about. And we're also looking at benefits sharing through the respective countries' Nagoya Protocol, and then looking at shipping customs, which many of you know can be problematic in sharing specimens. And then the third part of it is really building capacity at our 10 by 10 sites, so that we are providing reference material to them for molecular and serological testing capabilities, with partnering with the University of Marseille in Aix, France. And then we're developing sustainable business models. So what we also have here is that for some of the last part, capacity building, we provide reference materials on serological standards pegged to the WHO IS standards that Roger mentioned. We're looking at multiplex serology in this case, and multiplex serology is going to require the use of monoclonal standards, which most of the world has not, taken a deep dive with. The other piece that we're doing is that we have at our lab an ISO 17043 compliant site that we prepare reference and calibration materials in form of dry tube specimens, which we also have the long-term study, and they last for about 6 years at -20. So it's really good to have that available. I think I will stop here, and Thomas, you take over.
Yes, thanks, May. So the title that was given to us implied that we were going to talk about the link also between clinical epidemiological data and laboratory data. So between databases that cohort studies have and between the biorepository. So I want to briefly allude to that. I have some slides that are animated, so please click the next click. And so that talks about how we link the cohort platform. This is in this Contagio grant that was mentioned already, Work Package 1, with harmonized electronic CRFs. Please click again. That is the Work Package 2. Then with the whole area of ethical, legal, and regulatory challenges, Work Package 3. Please click again. And then also one more click for the virtual biorepository. So this is the project that I coordinate on behalf of Heidelberg University, and Colorado is a partner, and this biorepository is included here. Next slide, please. So why do we do this? Because in all these last years, we have been asking the same questions when faced with the next disease emergence. So what are the clinical phenotypes and their frequency? What is the frequency of severe versus non-severe disease? Are there congenital abnormalities? How transmissible? But then, of course, also, do we have biomarkers? What are the antibody kinetics over time? So please click again 2 times. Here we are talking about clinical data and laboratory data, and there needs to be a close link between them, which often is not the case. And that is one challenge we face. So please click again and then next slide. We are working with the European Molecular Biology Laboratory, EMBL. They have created, with our support, with Recoded and Contagio, 2 EC-funded projects, the Pathogens Portal and the Cohort Browser. As you know, the EMBL traditionally has been really focused on high-dimensional pathogen data in bioinformatics, and now we included this clinical epi part as well. So in the next slide, briefly, I can show you that, and please click again, these are kind of examples. There is the box that you see below there with V1, H2, and so on. Those are the different archives that they have. And one of them is in red, the sensitive clinical epidemiological data that has been added. And they're all linked via a common identifier now through biosamples. And that's our kind of pipeline for linking laboratory data and clinical epidemiological data. And that's the vision we are going to take forward. It's one of the examples we are working on. Next slide, please. So which barriers did we face? There is a lot of barriers, some experience here, and I don't have time to go into more details. Please click again. Those were two papers that were published, one with the example of the Zika outbreak and the research response, and another one, please click again, recently with sharing high-dimensional data and clinical epi data for COVID. And I'm happy to talk more about that and send you those. But I know that we have a little bit of a not so much time left. So please, the next slide, I will just talk about, click again twice. What we need also is this crosswalk between data standards. That is true for clinical epidemiological as well as for lab data. That's what we've tried here, inviting many of these representatives of community developed data standards, OMOP, LOINC, SNOMED, CDEX, and we've come up with this 5x5 table showing where there's already a crosswalk behind the scenes for interoperability. And that's really, really challenging, but very important that we work on that. And next slide. With that, I hope that we are just in time. I'm sorry that I was a little bit rushed. I want to thank everybody who contributed to that. Most of all, the VBS team, the Virtual Biorepository team at the Colorado School of Public Health and the Center for Global Health, but of course also the 10x10 members, the funding by the European Commission, and thanks for your attention.
Great. Thank you so much, May and Tomas. I'd like to wrap up really quickly by thanking all of our presenters today for sharing their insights, perspectives, challenges, and opportunities as they apply to biorepositories for pandemic preparedness. I'd also like to thank you for spending an hour of your time with us today. We know that there are many more discussions to be had in this space and look forward to hearing your questions, comments, and feedback, some of which we have already seen in the chat today. So thank you so much for that. We'll be generating a webinar report to share with you, and we hope to see you in the future when we plan to discuss additional topics in much greater detail. Be well, continue to do good work. Thank you so much.
Thanks.
Okay.